So what I found really interesting at the opening, I watched a lot of people who stopped by at the project, and it's a highly controversial one. So there was either people loved the project or people hated the project.
Really? What the project pointed out for me is how the general public are so far removed from how vital bacteria is, how common bacteria is, and how prolific bacteria is that they just have this idea that clean equals no bacteria. And it's like, no, life can't exist without that.
Well, I mean, if you look at beer and cheese, basically you've had lots of different bacteria for centuries.
Exactly.
I mean, now you obviously have the same starter culture that you use for different cheeses and different beers and things like that, but obviously ages ago you wouldn't have had that kind of sterility where you could only have one particular species or one particular flavor, so whatever you got for that cheese was what you got.
Well, I think that's it, and it's bread as well, and things like fermented vegetables like kimchi. It's like, you know, where did these staples come from?
You know, a response is better than just meh.
Yes.
So I think it was nice to see a lot of people kind of saying, oh, that's gross, and then I guess maybe the bit that's missing is explaining to people why it's not quite that gross.
Yes.
You know, I think people were actually surprised to hear that there was bacteria in cheese at all.
Yes.
Yeah. So I think thinking about, you know, fermented foods is a good place to start when talking about, you know, biochemistry or talking about biology in general.
Because, you know, cheese is, as we found out, is milk, rennet, salt, but it couldn't happen without bacteria.
But I mean, the coolest bit, I guess, with the sequencing, right, was that when we actually got the results back, was that we didn't really know what to do with them. You know, no one had really done this
no one knows
before, right?
So you don't know how it affects the flavor or anything like that. So I mean, it would be interesting in the future to then make a cheese and say we start off with this culture and how, like, what flavor does it have, and if we change this, what?
Yes.
It's hard to say if it's edible or not edible. It's actually quite tricky when it's something really food safe.
Well, and the cheese is not pasteurised. You know, it's not had those dangerous ones, and the best cheeses aren't pasteurised. So Tom, I think you should try it.
Yeah. Okay. Yeah, I'll have it. So. Yeah. Get in there, Steiner. Oh wow, that actually looks pretty good. What kind of cheese is this one again?
So that is a Stilton.
I see. Is it?
I guess like a unique version of a Stilton. I guess the bacteria is mostly outside, no?
I think we should try a bit of it, like. No. Whose bacteria is it? It tastes more cheddary, actually.
Better than the Tesco cheddar.
Yeah, would be hard, would it?
There's a nice rind. It has like a nice soft nibble.
I think it was a good first attempt.
I think so.